Welcome to my poster presentation. My name is Michele Fiscella. I'm VP Scientific Affairs at MaxWell Biosystems in Switzerland. And before I continue, I would like to thank Dr. Silvia Ronchi and Professor Andreas Hillman, who have significantly contributed to the generation of the results that I will present. So iPSC technology enables researchers to generate, for example, human neurons in vitro. And those cells can be generated with very highly reproducibility. Importantly, they can also be generated by patients, therefore enabling the modeling of human diseases in vitro, and they enable for high throughput experiments. And 2D and 3D cultures can be recorded. The electrical activity of neurons can be accessed. However, this technology created the need for high quality readouts to characterize the electrical activity of these human samples. I mean, one way to record the electrical activity for these human iPSC-derived neurons in both 2D and 3D models is high density microelectrode array technology, which enable the recording of electrical activity label-free and long-term. And in particular, high-density microelectrode array technology enables the characterization of single neuronal signals and also to extract, for example, subcellular details like axon. Today, I will present you the study by Ronchi et al., where the main goal was to characterize the electrical phenotype of multiple human neuronal lines. Here on the left, you see the fluorescence image of a culture, and on the bottom, the functional image of these neuronal cultures, and in particular, the 2D spatial map of firing rates. And on the right, you see how this firing rate activity evolves over time from motor neurons and dopaminergic neurons. And at the bottom, you see the quantification of the electrical activities from these two cell cultures and the comparison over development. And you can appreciate how high density microelectrode array enables the distinction of these two cell cultures across development. We've also studied the electrical activity of human neuron modeling neurological disorders like amyotrophic lateral sclerosis or Parkinson disease, and we compare the population spiking activity or this burst activity of human motor neurons to amyotrophic lateral sclerosis neurons and human dopaminergic neurons versus Parkinson disease neurons at 2 different days in vitro, 14 and 21. And again, you can see how high-density microelectrode technology enables the characterization and the separation of the electrical phenotype from these different cell lines across development, in particular by comparing the burst duration and the number of bursts per minute. As I've shown at the beginning, high-density microelectrode technology also enables to characterize and extract single cell and axonal signals. And here you can see how we could extract and characterize these networks in dopaminergic, glutamatergic, and motor neuronal lines. So we could record and automatically identify single neurons, track axonal arbors, and extract conduction velocity of action potential across multiple axons. Importantly, we also have studied the effect of retigabine, which is a potassium channel opener on motor neurons. And here you can see how the firing rate of these motor neurons could be modulated in a dose-response manner. In conclusion, I've shown you how high-density microelectrode array enables the label-free and long-term monitor of neuronal activity across multiple scales, ranging from neurons to single neuron to subcellular levels, and how we could use this technology to compare the functional phenotypes of brain disease models, and how to accurately detect and measure drug effects. With this, I would like to thank you for your attention, and I wish you a great ISSCR meeting.